just closed out. I would just close it and just open up his. Yeah, but you dropped his on the desktop.
You're ready with your next.
That one together because you don't really close that too.
You just lost the connection to it. Yeah.
When you open him up, what do you get? Is that it? That's it?
Yeah, that's it.
That's it. That is something.
I'm in the source again.
That's up to you. I don't
That's his
mind getting
presentation too.
Okay, everybody, it's 4:01. We are going to start in 1 minute. So please take your seats.
Yeah, good idea.
Check, check. One, two, 1, 2.
Okay, everybody, if you could please take your seat. We're about to get started. We're going to close the doors. Please grab a seat. Okay, everybody, I'm ready to get started. Could you please grab a seat? Okay. All right, we're going to get started with our next session. It's on clinical trials. And Dr. Alex Kalevzon from the Seaver Autism Center at the Mount Sinai School of Medicine is still traveling. Unfortunately, he's had a really bad travel day and his flights have been rerouted. He's not able to be here this afternoon. But we don't have Dr. Alex, but we do have his boss, Dr. Joe Buxbaum. Woo, yeah, yay! So, Dr. Buxbaum is a professor of psychiatry at the Mount Sinai School of Medicine, Icahn School of Medicine at Mount Sinai. And he is also the director of the Seaver Autism Center. And so many of you know about Dr. Alex and the work that he does at Seaver Autism Center. He sees patients and he involves them in natural history studies and clinical trials. And what you may not know is the other part of the work they're doing is kind of basic and translational research involving animal models, understanding the disease mechanisms of Phelan-McDermid syndrome. And Dr. Buxbaum has a whole team of people at Seaver who are working in the lab. So welcome, Dr. Buxbaum.
Good afternoon. Is that working? Sure. So I'll give you, so these are Alex's slides. I've made some adjustments, as you can see from the title slide. I've also taken out a couple of slides. That's me. I'm not the clinical. No. That's me. I'm the director of the Seaver Autism Center, not the clinical director. I've taken most of his slides as is. I've added a couple of slides because we got an email yesterday that I think is going to be, I was very excited about. And some of his slides about just the general kind of possibilities in clinical trials in PMS, I left out because I'm hoping to talk about that quite a bit tomorrow in the Friday afternoon session about how the field and how the families can go towards novel therapeutics. So I know that you've been told that tomorrow afternoon is going to be terribly tedious and boring, but if you're interested in kind of drug development, I will be talking a little bit about that tomorrow. So this will be primarily, yeah. Anyways, that's the end of my talk, and I want to thank you all for froze. No? In any event. So, well, we're almost there. Just briefly, I will talk about Igf-1 in Phelan-McDermid syndrome. And we do have a patent on Igf-1 and PMS, so I have a conflict there, so just take that with a grain of salt. Alex and I both have funding from Autism Science Foundation, from Simon's Foundation, from the NIH, so that might be a conflict too. He has support from Ovid Therapeutics, so those are our conflicts. No, they're all there. Oh, they were all there. So, oh, that's good. So the first, as Geraldine kind of mentioned, what we've been doing for the most part in clinical trials always starts from an animal model or from a basic science perspective. And one of the things we saw, early on, we were looking at our mouse models for PMS, was that the synapses, or the points of contact between brain cells, were a little bit immature. They weren't developing to the full degree. And it was already known in the literature and from prior studies that a hormone called Igf-1, insulin-like growth factor 1, is able, even if you give it as an injection, some of it goes into the brain and it's able to help push these nerve cells to a slightly more mature state. And the other thing we know about Igf-1 is that it's very, very safe. It's used a lot in kids. It's actually used only in kids to date for something called Laurent dwarfism. And so we were pretty sure that if we saw a positive effect in the animals, we could consider a clinical trial in individuals with PMS. And so we gave our mice, so these are mice where we mutate the SHANK3, the PMS gene, and then we study their behaviors and their kind of brain function. And when we gave these mice this Igf-1, their brain function improved and their behaviors improved. Right. So what Alex did next was just look at me. Just look at me now.
We're listening.
All right. If it's working. All right. Good.
PCs.
All right. So I'll come back to that. Well, let me start with this. So, one of the things that we've been very involved in Geraldine is really to be commended for kind of giving us a little push in this direction several years ago. There are something called RDCRNs, Rare Disease Clinical Research Networks, which are multiple sites that work together around rare diseases. Mustafa Sahin is an expert on tuberous sclerosis, and he was working with somebody who's an expert on P10 syndrome, another rare disorder. And Geraldine convinced them and convinced us to add PMS to the grant. And so that consortium is called the DSC. And as you can see, PMS is a very significant part of it, and actually one of the more interesting parts of the consortium in some ways. And what we're doing there is characterizing PMS extensively using medical, behavioral, and cognitive measures, doing the natural history study that you all know is so critically important, and looking at biomarkers that we can measure things very accurately so that we can see small changes over time to see if we're going in the right direction. And then the consortium as a whole always has a component that's around novel therapeutics. What might be done for these rare disorders. So that's actually going back in. It's going for renewal at the end of this year, and we're talking a lot about what we need to add for PMS to make, for the next 5 years of research. And this consortium is very, very much clinical trial ready. So many of the people in this room who are on the research side are involved in this consortium. Craig is, Liz Barry-Kravitz, Latha Surya, I'm missing some people. Audrey Thurm, I know she's here, she was here. And so the whole network is using similar measures, and they're all very skilled clinicians and clinical trial specialists, and so we are in a position to do clinical trials across multiple sites, which is critically important. All right. So these are some of the trials that are going on at the Seaver Center, which will be my focus. Many of the RCTs are still at the Seaver Center. This is what I talked about earlier. We take a mouse and we genetically modify it. So this is like the Franken food. This is Franken mouse. So Franken mouse has a mutation in the PMS gene, and we can look at things like brain function. This is a measure of how well cells talk to each other. And when you're missing one copy of SHANK3, this orange line down here, there's some degradation in the signal. But we can reverse that with this compound called Igf-1. And then this is a behavior. This is how long it takes a mouse to fall off a rotating rod. So it's actually a very easy motor test. And you can see that this is a typical mouse, and the mouse with a SHANK3 mutation falls off more rapidly, but if it got Igf-1, it does better, right? So that's kind of the basis. This was quite a number of years ago, 2013, we published this. But that was the basis. That was the basis of this Igf-1 trial, which I think you've all heard about, but there's some news I want to share. So Igf-1 is a hormone. It's a growth factor. It binds to a receptor. This is like a cell membrane. It diffuses into the extracellular space. It binds to a receptor, and it activates some internal signaling cascades. So it changes the function of the cell. The trial that Alex first did was a crossover. So it's a clinical trial. It's random, so everybody who walks in the door is randomized to either placebo or to Igf-1. They get it for 12 weeks. There's a washout period, and then they're switched to the other arm. So everybody gets active drug sooner or later. We just don't know which arm you're in. And it's not necessarily a fun trial because you have to get injections twice a day, and because there are problems. There is a small risk of hypoglycemia with this treatment. There's very frequent glucose testing. All right. So in this first, and it truly was a pilot study, the main outcome measures were social withdrawal on the ABC and repetitive behaviors in the RBS. And both of those outcomes, the drug showed significant improvements. So higher is better, so that the ABC got better, and higher here is also better. There was reduced repetitive behaviors. Okay? So it really was a trial, it was a pilot, it was an N of 9. And why did we only do 9? We didn't have that many patients at the time, but also the drug cost us about $14,000 a patient. And our hope and plan and expectation was that the company that makes the drug would be excited enough to help us all with a free drug for a clinical trial. The NIH got excited about this, and they funded a follow-on trial that was supposed to be another 20 patients, but because of changes in drug price, it actually went up in price. We've done another 10. So this is what I showed you before for repetitive behaviors. This is the original study. Well, look down here because this is corrected. So in the original study, there was a positive effect of the drug on repetitive behaviors. In the follow-on study, there was a trend in the same direction, and when you combine them, you had a significant finding. Okay, so there was a replication of the repetitive behaviors finding. Unfortunately, the social withdrawal scale did not look so good. So this is the first study that I showed you. This is the second one. This is the combination. However, 2 other measures looked very interesting looking across both trials. One was hyperactivity. So this is the first trial. So getting better again, but trending. And then the combination of both studies with 19 patients also getting better. And another thing we saw that a little more complicated was the sensory issue seemed to get significantly better as well. We only tested that really in the second trial. So we're pretty excited. We're like, there's still some positive things going on. We go back to the company, and the company's name is Ipsen. Don't boo. I know many of you know that we've been discussing this with Ipsen for quite a few years. They just got back to us. Oh, let me show you where. Just this on July 18th of 2018, which is yesterday. And our proposal for a larger clinical trial has been approved by Ipsen. It was approved on the 17th, but they emailed us on the 18th. What can you do? So that trial, the proposal to Ipsen, and they will, I think they pretty much accepted it as is, but there'll be some conversation, is to, as always, make sure it's safe and tolerable, to look at efficacy in the social withdrawal, which I've already shown you, efficacy in the repetitive behaviors, but now also adding sensory reactivity, because we saw some action in the second study, and hyperactivity, where we saw positive changes in both when we looked carefully. And then we also have, this is one of those biomarkers I talked about, which is a way of measuring things very accurately so you can see small changes. And if you see a small change in 12 weeks, it makes you want to do it for longer, right? So, and the reason we did that is because this is an EEG called the visual evoked potential, and we had 6 patients that we were able to, as part of the second trial, we had our EEG in place. We were able to measure their EEG pre and post treatment. And what you can see is something called gamma power actually improved in most patients, most individuals with IGF-1. We don't know what that means yet, but if it's a predictive marker that things are going in a good direction, it's a way of kind of, as I said, if you see something moving forward in a positive direction when you do 12 weeks, then you say, okay, let me try it for 6 months, right? So we have to talk to Ipsen. They have to review all the paperwork one more time, but we fully expect that this trial will begin. And the funding we requested was for 50 patients in total, 50 individuals in total. This is now going to be a classic 2-arm design, so half the individuals will be on placebo and half on active drug, but everybody will be on an open label extension. So everybody gets drug here too. Why do we care about that? Because you don't want to, nobody wants to stick their kids twice a day and then maybe just be on placebo the whole time. All right, so this is another design that's very common, where if you have more money and more drug, then everybody can be on the open label extension, so everybody gets access in the second phase, but they're randomized in the first phase. All right, so it'll be 50 kids, 2 to 12 years old. Obviously, well, I will tell you that they have to their bones still have to be growing, right? So they can't show signs that their bones have stopped growing. So anyways, that was really a multi-year struggle, and we're very happy about it. And we do a lot of stuff, but I showed you the main outcomes we're going to be looking at. All right. The other clinical trial that's ongoing is oxytocin. Oxytocin is called the love drug. And the reason we got involved in oxytocin was also because of our animals. These are actually rats, and these are rats, again, they're Frankenrats. We've mutated the SHANK3 or the PMS gene in these rats, and they show very interesting changes in behavior. One thing they do is they don't do a good job of remembering rats they met yesterday. So rats are social creatures, and when you put a rat together, the white and the black rat together for 1 hour, and then you take them apart and you put them back together. So there's the white one, there's the black one, but you add a new rat that nobody's met, a gray rat. The test rat will spend more time with the new rat. So rats, like many social hierarchical animals, they go and check out the new person and they kind of interact with them. So a typical, I don't have the typicals here, but a typical situation like this, you'll see much more time spent with the new rat versus the old rat. But in the SHANK3 rat, the PMS rat, it forgot about this. It no longer remembers a social recognition memory. However, if it gets oxytocin, then it looks like it's supposed to. So there's a deficit in remembering social events, and if you give them oxytocin, the rats perform as typical. These rats actually also show deficits in attention, and as I think you know, attentional deficits are a big part of the syndrome. We can measure that by asking the rats to pay attention to little flashing lights on a screen and to hit them and to kind of like remember, like they have to wait, hit the screen, go get some food, so you can kind of see how much they're paying attention, how often they make mistakes, how often they're kind of impulsive and jump in before they should. And here too, the rats with the SHANK3 mutation actually don't do so well, but with the oxytocin, they get better. So their attention goes up. Right. Oxytocin, again, is an incredibly safe compound. We've worked with it at the Seaver Center for more than 10 years. And so here, and it's also much cheaper than IGF-1. So here we could do the same design I showed you, placebo versus oxytocin, and then an open label extension. So we're recruiting for that right now. And the final trial, and the procedure's pretty much the same. We have our primary endpoints around social behaviors and things like that, but we also get, and this is sensory information and so on, but we get a lot of data. There are side effects for all of these compounds. This is potential side effects for oxytocin. And I will jump now to the third trial that's ongoing. I think, as you know, AMO Pharma is one of the sponsors for this symposium. Joe Horrigan, I know, is here because he was up. I saw Michael Snape's name on the list. I don't know if he's here or not. No. Well, but Joe's here. So I'm going to talk about the slides that they talked about with Alex, and Alex then turned over to me, and I'll make some errors, but then Joe can correct me. So this is about a compound called AMO-01, and it is to look at its efficacy as an anti-epileptic in PMS. So this compound, AMO-01, is an inhibitor of what's called the MAP kinase, or the ERK pathway. So again, this is a cell, this is a receptor, and there's these signaling inside the cell that turn things on and off, and this compound reduces that activity. In prior studies, preclinical studies, AMO looked at some of these mice I talked about, and they asked questions about whether the ERK pathway, this ERK pathway, is elevated in SHANK3 or PMS, and it is, compared to other signaling pathways, both in the brain and in the blood. So this is a measure of how much is there excess ERK activity. So, we have many, many enzymes in our body that do all sorts of important things, and so this particular kinase or enzyme is overactive in the mice missing a copy of SHANK3, or both copies of SHANK3, and both in the brain and in the blood. And their drug turns this down, right? Does that make sense? So, so they took their compound. Oh, come on. It was like 20 minutes just waiting for the. Yeah. Give me an extra. I need an extra. These aren't my slides. I'm reading them as I go. All right, 30 minutes. Got it. So they gave their compound to these mice, and the mice showed some of the behavioral things these mice were showing before: increased anxiety, excessive grooming, impaired social recognition, sensory motor dysfunction, species behavioral deficits, and seizure thresholds, all turned around by the treatment by this drug. And the seizure thresholds, these are audiogenic seizures you can induce with certain kinds of sounds, you can induce seizures, and so their threshold went down. They become less prone to seizures. Okay? And seizures, they're a very significant part of the phenotype, as you know. They're also relatively easy to quantify, right? You can say seizures are going down or not. So this is the study. Make sure it's safe and tolerable. It's a 6-hour IV infusion, and then look at reduction of seizure frequency, and just overall measurement of efficacy in a clinical perspective. These are their inclusion criteria, exclusion criteria. Jordana Weissman is a study coordinator. If you want to write that down, if you're interested, you can reach out to us about any of these studies, but particularly the AMO study. And I will stop there because I know what the shepherd's hook looks like. This is the Seaver Center and Alex, who did a lot of the work, and a lot of the preclinical work is over here under preclinical. And this is the RDCRN DSC, and we really have a great relationship with, as I said, the larger consortium, some people I mentioned, the neuropsych group, and the leadership in Mustafa Sahin. Done. Right on time, apparently.
Thank you, Dr.
Awesome.
Buxbaum.
Yeah, and I'll just add, it's really been wonderful to see all these different researchers from different institutions working together to answer some of these really key questions about the natural history of Phelan-McDermid syndrome. And most of them also have some other initiatives also going on where they're looking, trying to understand the biology of Phelan-McDermid syndrome. So they bring that expertise as well. So it's really been a wonderful thing to be involved with, and we're very grateful to all of the researchers working together in that. So we're ready to put up a poll. Julie said it. Yep, it may not be showing, but it's on your phone. So you can answer the poll and then get started with your discussion. In case you're wondering, what do you talk about with clinical trials, since that's not like a daily thing that you're worrying about, some questions that you might start with are, have you participated in a clinical trial? If you have, why? If you haven't, why? What are some of your concerns about participating in clinical trials? So just any questions along those lines.
There was somebody that recently asked if we could add regression to this, and unfortunately we cannot. These are the only things that we're voting on now. But please feel free to make that part of your conversation and add to your questions. Joe Horrigan. Are you here, Joe Horrigan? Just a quick reminder, we have 5 minutes until we are going to reconvene for our panel. So if you have questions, please start to input them and feel free to vote up and down if the question is applicable to you. Thanks. In a few minutes, we'll be reconvening, so please get your questions up and or upvote or downvote any questions applicable to you.
think we're getting our panel assembled. Okay, if we could ask you to please wrap up the discussions around the table.
Okay, we're going to get started.
All right. Great. Thanks very much. And I can tell this is a really spirited discussion. I hear a lot of good conversations. We had some great conversations at our table as well. And we've got a great representation in the panel. Just by means of introduction, for those who don't know me, my name's Kris Munroe. I'm a neuroscience researcher. You can't hear me? All right. I'm also known as being very quiet. So my name's Kris Munroe. I'm a neuroscience researcher. I've worked for a couple different companies. The current company I'm working with does work in the rare disease space. In my day job, I am part of clinical development teams, and that's the team that takes a new drug and will move it into clinical trials all the way through different stages. So that's sort of my background. We have great representation on our panel here. I'm also a PMS parent. My son Jonathan is 15 years old, so I have a vested interest, I guess, in hearing what's going on here as well as people do in the room. So just maybe what we'll do is just quickly go by means of introduction. I think you've met most people, but I think in terms of clinical trials, maybe talk about what your background is there and that relevance, then we'll step through the questions.
Okay, I'm Liz Berry-Kravis. I introduced myself before. I guess I could be referred to, I'm a clinician researcher at Rush and have been working in translational neuroscience, in other words, trying to bring discoveries in neuroscience to families with developmental disorders now for really since 2002, when we first started doing that in Fragile X. We've run over 20 Fragile X trials. We've got 5 Rett trials going on. We're running a couple of Angelman syndrome trials. And we've done trials in various neurodegenerative diseases as well. So we've got a lot of clinical trial experience, and I think the Fragile X field probably has the most trial experience of any single gene disorder that is a neurodevelopmental disorder, because we've had a great number of failed trials because we started doing this really early. We probably followed some wrong models, and so we've learned a lot from that in terms of trial design and what we can and can't do, and we're hoping to be able to apply that knowledge to sort of all the other fields that are following us to kind of do that work.
I've been involved in 3 clinical trials. I treated the mice with Igf-1, the rats with oxytocin, and some other mice with actin modulators. And so I'm a preclinical scientist, obviously. But the work we do is always with clinical trials in mind.
I think this one's working. So I am not a clinical trialist by training, but I've worn quite a few hats along my professional arc, if you will, all of which have placed me more in a strategic sort of executive oversight role over many different clinical trials and clinical portfolios of clinical development activities. I headed the autism unit at Pfizer many years ago, when that was really the first dedicated unit of its kind, trying to figure out how you turn science into medicines for not just autism, but the array of genetic syndromes that make up the spectrum, and oversaw everything from target ID to proof of concept trials there. And as a chief science officer at Autism Speaks, was really responsible for helping shepherd a portfolio of funded clinical work, small studies in a variety of different modalities, not just medicines, but technologies through our venture arm and a variety of other things. And I work now primarily as a consultant. Principally, my clients are large drug companies and technology companies, some venture funds, all of whom are very interested in autism, but really interested in it through the lens of rare CNS disease, principally examples like Phelan-McDermid, where there really is tractable biology out there. So in many ways, I'm helping the industry or the marketplace of clinical development organize their strategic investments in it. So I've become a bit of a consumer of what clinical trials, the role they play in helping move the entire story forward. So I will be able to at least offer some insights from those perspectives.
Right. Well, thanks very much. I think we have, so I think this question of getting involved, yes? Okay, yeah, is that better? Yeah. So I think the first question really is about getting involved, and I think what I would actually add on to that is finding out about these trials. I think that's probably something that we heard from a few different people. So maybe if your perspective is about finding out about the trials, number one, and then becoming involved.
I would jump in right here, and like many small foundations, can you hear me now? I can't encourage you guys enough to get involved in whatever registry activities are going on at the foundation. The investments foundations like PMSF are making in what we call clinical trial readiness. That is, once a company just finds that they've got a molecule or a medicine that may have been developed for cancer or diabetes, suddenly might have a connection with the Shank3 story, the interest is to get into clinical development as soon as possible. So they need to find and recruit individuals from your community as fast as possible. Registries offer a digital surface area, if you will, with the community that can be accessed right away. So not just registering yourself, but providing as much information about your child's clinical journey to what we call annotate their identity in the registry is going to help, I think, in enormous ways. So if you're not in the registry, get involved in it. I can't encourage you enough. That's what I would say.
So every clinical trial in the U.S. is posted on clinicaltrials.gov, and if you just search PMS, you will find it. There's only a handful that I think I mentioned them all at this point. But the foundation itself, the newsletter talks about some of these trials. I know that AMO itself advertised, and the newsletter will include the AMO trial. As we go forward with the Igf-1 trial, we will definitely, Geraldine will know. It'll be in the newsletter. So that's the best place for what's actually going on in PMS.
And also I was going to note that families who are in the registry, when researchers come to the foundation and say, hey, we'd like your assistance with finding patients that might meet criteria for our research study, that goes through the registry. So if you want to find out about all of the opportunities available to you, you want to make sure that your child is registered. If you're not registered, you may not always hear about the research studies that are going on.
Liz, anything to add?
No, I think once a trial starts, it's really important for whatever disease it is for the foundation to post the information on that. And generally, the sites will be posted. And usually trials, we're looking to enroll patients. So if there's a coordinator at the site, you call the coordinator. And if you're anxious and interested in getting in right away, usually you can get scheduled pretty quickly once the trial is up and running.
Yes?
The AMO study is looking, they have some open slots. We're in it, and I know that they're looking, and I just...
Yeah, so AMO has already started, as you know. I put up Jordana's email address and contact, but I put up contact information for Jordana if somebody is interested in hearing more about the study, or you can email me or Alex. Yeah, we are recruiting, and as Geraldine mentioned, we're doing it with her help. Not easily. Do you remember how long it took to get it up the first time?
Maybe they're not the right person to ask, right?
Kris.
All of the live streams will be up on the website afterwards, so you can go back to that.
But just please come up to me, and I'll lend you my computer to look at.
Yeah. Okay, and so let's move on to the next question. This is really actually a good one. I think it's how do we know if the improvements last past the end of the clinical trial? And I think that could be expanded a bit too. How do you get information about, you participated in a trial, that sort of stuff afterwards?
So for most of the small molecule drugs that we're talking about, working on a pathway in the brain, Probably when the drug ends is what we expect when the effect ends. Sometimes there's a little bit of what we call carryover effect, which means the effect of the drug lasts longer than when it's actually in the body. So we expect for a short trial, if you can't stay on the medication and you saw a benefit, for instance, or you thought you saw a benefit, that's not expected to continue. An exception to that is certain things like gene therapy, where you may only have to do it once, or maybe a booster later on, so you don't have to keep administering the drug. But the idea of the trial really is to develop the data that's needed to go from one level of trial to the next level of trial, and then ultimately to have the drug approved so you can just stay on it all the time. So that's really what the process is. And then in terms of follow-up, generally after the trial there'll be announcements about what happened in the trial. And like I say, when you start with a phase 2, with an early phase 2 trial, your goal is to be able to go to a larger phase 3 trial, something that will be, quote, a registration trial, and then the FDA will be able to base an approval on that. Sometimes with super rare diseases, you can combine phase 3 and phase 2 and phase 3. And I think I'm approaching the how long does it take question also. So I think it depends on what kind of trial you're doing. If you have a successful phase 1 or 2A trial, you're still going to have to do more trials. So then that's going to, be a number of years further. But I would say if things go really well and it's pretty successful, you're still, talking about a 3 to 5 year process. I don't know if Rob would agree with that, but I think that's usually the situation. Sometimes when a company is up to phase 3 in a trial, then they have to accrue safety data. So they'll offer something called an open label extension. And the trial, the IGF trial, is having an open label extension at the end, but it's only, it's only 12 weeks. Whereas sometimes when the company really thinks they're going to get this approved and they're doing a phase 3 trial, they'll let you stay on the drug until approval happens. But you have to go through all these steps, and so it does take some time.
And I would just only add to that, one of the benefits, just from an education point of view, there have been several pieces of legislation that Congress has passed many years ago in some cases that have incentivized companies to work in what we call rare diseases, orphan diseases with less than 200,000 people in the United States, which the Phelan-McDermid syndrome community easily qualifies. And with that incentivization is not necessarily a relaxation of the safety requirements, but it does put in place some things that accelerate the process of clinical development. So you do have the potential to move clinical trials faster, but it really requires patience. It's a process intended not just to generate evidence that you can count on that something is working, but that it's actually safe. And that's really, I think, the world we find ourselves in. I'm sure you see all sorts of treatments and various types of interventions promising miracles which don't have evidence out there. This is a process that's really intended to build confidence in what comes out there, and you're not just a subject in the process, you're a partner in this. I think to that second point that you really have an opportunity to teach us what is happening with this medicine, this experimental agent, and we don't know before we start whether or not improvements are going to last. We don't know a lot about it. That's why we do clinical trials. You are a partner in this process, and it's your insights into how something's working in your child that teach us the answers to those questions, even if they weren't built into the design of the study. So you have a very powerful, enfranchised role in this whole process. You're a partner in it. I think that's very important for you guys to appreciate that.
I think it's also important to ask questions about the trial. If you're interested in it, again, this is your child, it's your life, and really you should be asking and comfortable with participation, and ask as many questions as you need to make sure it's right for you.
And I'm discovering that I'm a very, very concrete person because all my answers are very concrete. So with the Igf-1 trial, there was very little evidence of carryover or carry forward. And at least with the VEP treatments, it looked like after a month or so after cessation, things were going back to baseline. So there wasn't evidence of persistence, and that goes to the point that Liz was making that, as I mentioned before, IGF-1 can be used for 6 or 7 years in kids with Laurent dwarfism. We have one of our parents here whose child has been on IGF-1 for 9 months and then started again. I don't know if that parent wants to talk about it. We do require longer trials, but the sense right now is, at least with these 12-week trials, there wasn't a lot of sustained positive things, at least by the small measures we made. That doesn't mean there weren't, it was just we couldn't see them. And just to echo, we're involved in a trial with another rare disorder where basically we're pretty sure that the FDA will give us a one and done, an adaptive one and done design. In other words, we're going to be able to, it's through a CRO, we're going to be able to do a single pivotal trial that actually can change as it goes forward in an adaptive way, and that will be sufficient to get approval because it's a rare disorder. So there really are some very, very profound positive implications for rare disorders, and biotech and pharma appreciate that because because it brings more money to them, but also because they get better patent protection. It's a much easier regulatory pathway. So I think we're closer than we think if we have a good drug.
Yeah, I'm involved in a trial like that also in a rare disease called Niemann-Pick type C, and we're doing a phase 2-3 trial in the FDA. We have a thing called fast track with the FDA, which also means that this looks like a therapy to the FDA that could be very likely to succeed, and so every time we send something in, they have to review it faster. And so that does speed it up. And basically, the approval, if this goes through, will be based on a trial in 51 patients. So the FDA is looking at some of these new designs in rare diseases. They're interested in rare diseases, but we've had a lot of meetings with the FDA. I've been there in 3 different divisions for 3 different diseases. And uniformly, what they want is they want to understand, because rare diseases, there isn't a lot of literature out there. What they want to know is that your drug is improving something that is important to the families and the patients and is hooked into what they view as their quality of life, and they call that voice of the patient. And what you were doing right before this, when you were voting, is exactly the information that the FDA wants for every rare disease. We're doing it in Fragile X. They're asking us this in every condition. So we want to be targeted towards something that we can show the families think is important.
So I think in terms of the question on signing up for the IGF-1 study, maybe again, that's if people are interested, you could talk to Dr. Buxbaum. The medical marijuana, I think, Liz, we had a brief conversation about that.
Yeah, I've been working there are a number of companies that are developing medical marijuana. GW, most notably, a company in the UK just got Epidiolex approved for epilepsy, and it had 3 controlled trials that showed that it is effective for epilepsy. It's approved for epilepsy, so if you had PMS and epilepsy, there would be no reason you couldn't use it for a PMS patient. It doesn't have to be specifically approved for PMS, but you would have to have epilepsy. So there's another company called Zynerba that we're also working with. They're working on Fragile X. GW was working on Fragile X. I think they may be moving to Rett syndrome, but there is a high interest in these companies in neurodevelopmental disorders, and does this drug help with calming, anxiety, agitation, irritability, aggression. So these are in talks, and kind of what they're doing right now is kind of going with the disorders they think might be the lowest hanging fruit in terms of getting an approval. So I would see I think if there's an approval in one specific developmental disorder, then these companies are going to try to rapidly expand this to a more general indication. So I think there will be work in this area.
Right. I think we have time maybe for one more question. I know one that came up twice was about clinical trials for adults. So maybe if we could get your comments on that quickly.
So I'll just say the AMO trial is to age 45. And so the epilepsy part is for adults. The IGF-1, because the kid, you have to still be growing to under the current current use of IGF-1. The IGF-1 trial is restricted to people who are still growing, but the AMO trial is until age 45. So I think adult trials will go on, but they'll have to be a kind of a proper relationship between the outcome you're looking at, the drug you're using, and so on.
Yeah, actually, most of the time, if it's a totally new drug, which is not the case for IGF-1, the trials have to be done in adults initially, and we have to get safety data. And we've been struggling with the idea that sometimes companies, if there's not enough movement in adult trial, then the company won't go and study kids, so that we think might be more pliable developmentally and might be more likely to show a response. So it's actually usually, you just happen to be in a situation where it's not that way, but that's usually the situation. So I think as new agents come forth, you're definitely going to see adult trials.
So,
So, as I said, we went with IGF-1 is currently approved for a specific indication, which has to do with growth. And so rather than go to the FDA and go through this long process of saying we want to use it outside the currently accepted range, we said we're going to keep within your currently accepted range. That's why kids had to be growing. I feel comfortable, maybe because I'm too optimistic, that if there was evidence of efficacy, one could then go back to the FDA and say, okay, we're not using it for growth. We want to use it in a way that's not been used before. Let's go through that process. And obviously, in the fullness of time, if IGF-1 is a possible approach, we want to replace it with small molecules or things that are much easier to administer, don't require twice-a-day injections, and so on. So this is just, you know, a compound that was very safe, that was out there. We knew we can get it into kids pretty quickly. It had some interesting evidence in the mice, so we did it. But it doesn't mean it's the optimal thing.
Right. I think the issue is we expect the IGF-1 and PMS is having an effect on the brain, and the growth effect is maybe a little bit different and has to do with the body, but it just is creating a limit in terms of what the age range is. Whereas the effects on the brain should not be something, you know, in PMS should not be something that goes away necessarily, unless the adult brain is just less movable because, you know, it's spent more time wiring.
Yeah, but it's not so hard to go back to the FDA and say we're going to do something different than what's actually already been approved.
So I think this is a fantastic sort of, I think, format. As we heard from many of our speakers, McPosium really puts families and kids and researchers together. Researchers really like to talk about their work, so I would encourage you to track them down and talk about. They'll talk forever. The only people that like to talk more are parents about our kids, and maybe actually grandparents about their kids even more. So it's, I think, a really nice format and would encourage that interaction. And I think with that, I'll ask you all to thank our panel members and presenters today.
Great. You know, this